A very good morning, all of you. Today we'll be talking about gingival crevicular fluid. So, as the name itself suggests, that it is a fluid which is coming out of the gingival crevice or gingival sulcus. Therefore, it is also called as gingival sulcular fluid because the location from where you can collect GCF is your sulcus. So, these are the contents of the discussion today. Introduction, history, GCF production, methods of collection, composition, GCF as a biomarker, clinical significance, and conclusion. So to start with, GCF is a physiological fluid and or an inflammatory exudate that has been recognized for over 100 years, which provides a unique window for analysis of periodontal discussion. So how did they found out that in what conditions it is present, how much can it be collected, and what does it contain? That should be the highlight of today's lecture. Let's talk about gingival sulcus first of all. Gingival sulcus, as I already told you, that it is the space. It is not a structure. It is the space between tooth and the epithelium. Therefore, it can be defined as a V-shaped shallow crevice or a space around the teeth, which is bounded on one side by the tooth and on the other side by the epithelium, lining the free margin of gingiva. So clinically, how can you evaluate the depth of gingival sulcus? You can evaluate it with the help of a periodontal probe, which has markings on it, and the estimation can be done by the distance which it penetrates the gingival sulcus. So under normal conditions, this depth is usually 0 millimeter to close to 0 millimeter in a newly born child or in notobiotic rat, which are free from microbial load, which are free from any kind of oral microflora. There you can see a 0 millimeter sulcus, that is it, they have no sulcus. But at the same time, when eventually the oral cavity it is inhabited by the bacteria and there is an increase in sulcus depth in histological sections, usually a variation of 0 to 6 millimeter was studied by Auburn and Kohler, and an average of 1.8 millimeter of sulcus depth was given. Other studies have reported 1.5 millimeter of sulcus depth and 0.69 millimeters. The histological depth of a sulcus does not need to be exactly equal to the depth of penetration of the probe. The so-called probing depth of a clinically normal gingival sulcus in humans is 2 to 3 millimeter, and pathologically deepened sulcus is called as periodontal pocket. So talking about the history of GCF, to begin with, initially in the year 1950s, there were two scientists by the name of Brill and Crass. What did they do? They injected dogs intramuscularly with a dye named as fluorescein dye. And within 3 minutes, they introduced a filter paper into the sulcus of dogs, and to much of their surprise, they saw that the same dye could be recovered back from the gingival sulcus. So what did this discovery lead to? This discovery led to the finding that fluid from the bloodstream passes through the tissues and exits via gingival sulcus in the form of a gingival sulcular fluid, or the GCF, which we are talking about. But initially, Brill thought that GCF is only some kind of normal physiological fluid. It is an ultrafiltrate of blood. He termed it as a transudate. However, uh, it was Low and Home Pedersen only who considered it as inflammatory exudate. They demonstrated that GCF is an inflammatory exudate and not a continuous transudate. In a strictly normal gingiva, very little or no fluid was collected. Only in inflammation condition or irritation condition can this fluid be collected. So how to define GCF? We can say that GCF is defined as a physiological fluid secreted in the gingival crevice that is classified as inflammatory exudate during disease or serum transudate during health. So many times you often get questions like in MCQ based question, you often get this question that GCF is a exudate, transudate, both of the above or none of the above. So the correct answer will be both of the above. Yes, it can be considered as transudate. That is also correct. And it can also be considered as exudate depending on the healthy and unhealthy condition. Healthy that is, there will be no or very minute amount of GCF, and in unhealthy, which is an inflamed condition, you will find quite a good quantity of GCF there. Okay, so talking about the formation of GCF. How does this GCF form? There were two scientists, Alfano and Pashley. Alfano in the year 1974 and Pashley in the year 1976 suggested that the initial fluid is the interstitial fluid only, which is the physiological fluid, which appears in the crevice because of the changes in the osmotic gradient. This initial pro-inflammatory fluid was earlier considered to be transudate, and on stimulation, it changes into exudate. So if we talk about Pashley's model, Pashley suggested that GCF production is governed by the passage of interstitial fluid from the capillaries to the tissues to the lymphatic system. These are the capillaries, this is the lymphatic system. So how will be the interstitial fluid flowing? Interstitial fluid will be flowing from the capillaries to the tissues, and it will be it will be taken up by lymphatic system. But when the capillary filtrate exceeds that of the lymphatic uptake, where will this fluid go? This will accumulate in the form of edema, and this will leave the sulcus in the form of gingival crevicular fluid. So if we talk about non-inflamed or healthy condition, we all know that there is less vascular permeability, and therefore in turn there will be low GCF flow and high uptake by the lymphatic vessels. But during inflammation, what is the cause of inflammation? Plaque, and plaque harbors bacteria or microorganisms, which creates a osmotic gradient. There will be increased leakage of protein molecules. The increased hydrostatic pressure will result into increased vascular permeability, and ultimately increase in amount of GCF as well. So talking about the functions of GCF, first of all, it causes clearance of dead cells and bacterial molecules from the sulcus. It exerts antibacterial and antibody activity due to the presence of different immunological factors. You can also appreciate immunoglobulins, lysozymes, inflammatory cells in GCF. And if somebody asks you, and this is yet another MCQ based question, which is the most predominant immunoglobulin found in GCF, the answer is IgG. Okay, so just to summarize the functions of GCF, we can say exerts antibody activity, it cleanses the sulcus, it possesses antibacterial activity as well, and one of the most important function which you often miss at is that it contains plasma proteins, and these plasma proteins are helpful in causing adhesion of the epithelium to the tooth. Now, next is method of collection of GCF. As I've already told you that very little amount of GCF can be seen in non-inflamed condition, and slightly more GCF can be seen in inflamed conditions. There has to be technique sensitive, expertise to collect the GCF, because it might get contaminated with saliva. It is not at all easy to collect GCF. So there are specific techniques for collection of GCF, like absorbent paper strips, twisted threads, micropipettes, crevicular washing. Let's talk each one of them one by one. First of all, the absorbing paper strips, usually by the name of Perio paper. These strips are available commercially. The name is Watman's Perio paper. And there are two methods of collection: intracrevicular method, which was given initially by Brill et al. Brill has done a lot of studies, initial studies on GCF, and again, Low and Home Pedersen also have contributed to a lot of studies related to GCF. Let's see how. Brill in the year 1958, he gave the intracrevicular method. Intracrevicular, name itself suggests that the absorbent paper strip or the filter paper strip, they are introduced within the sulcus. If they are introduced within the sulcus, what will be the drawback? The drawback is they will be unnecessarily inducing some kind of irritation to the sulcus. If they are irritating the sulcus, then there will be stimulation of GCF flow. If you yourself are stimulating, how will you be able to assess what is the normal flow rate? So here is the major drawback of using intracrevicular technique. Later, Low and Home Pedersen minimized the irritation of this method given by Brill et al. And placed these absorbent papers just at the entrance of the sulcus. And since on the entrance of the sulcus there is no question of disturbing the sulcus or irritating the sulcus, so you can easily assess the exact amount of GCF. Okay, so you can see in this photograph that how you are placing the how you are placing the absorbent filter paper strip, the perio paper, you're placing just at the entrance of your gingival sulcus without irritating the sulcus. So this method definitely has an advantage over the initial intrasulcular method. Okay, so let's talk about the advantages and disadvantages. The major advantages: readily available strips, and it is very easy to use, can be applied at individual sites, and yes, it is least traumatic when used correctly. But at the same time, what is the disadvantage? Disadvantage is, here in the slide you can see the amount of GCF collected within 3 minutes is just zero point 0.1 milligram. So there have been experimental studies which suggest that about 1.5 millimeter width of filter paper, if inserted 1 millimeter within the gingival sulcus of slightly inflamed gingiva, absorbs about 0.1 milligram of fluid, and that too in 3 minutes. So you can understand that the amount of fluid is very minute. And again, this fluid can get contaminated with saliva, and there are chances of evaporation as well. Second method was given by Weinstein et al. Weinstein et al. Used pre-weighed twisted threads. So you can imagine these twisted threads, if I'm saying pre-weighed, they are weighed before the sampling is done. Okay, and immediately after collection of GCF, they are again weighed. For example, if before or prior to the collection, the weight of the thread was, say, 22 micrograms, and later it becomes, say, 24 micrograms, so the collected amount will be 2 micrograms. I'm just telling for example. So the difference between the two will give you the estimation of GCF collected. The time of collection was measured with the help of a chronometer, and this enabled the investigator to express the flow of fluid as well in milligram per minute. And what was the amount? We just discussed the amount was 0.1 milligram, and that too in 3 minutes. The width of the paper, of filter paper, was 1.5 millimeter, and how much depth of the sulcus was there till which it was introduced? It was 1 millimeter. Next method is micropipettes. Usually collect gingival crevicular fluid by the force of capillarity. You can see this is a micropipette. We have introduced it within the sulcus, and by the capillary action, the fluid will be collected, and this fluid is later centrifuged and analyzed. Advantages: it provides an undiluted sample of native GCF. We'll be talking about the diluted sample as well. The next method is crevicular washing technique, whereby we collect a diluted sample. Here the major advantage is that it provides an undiluted sample of, means whatever concentration of GCF, of native GCF there, the same concentration is collected. But it has a major disadvantage that it has a prolonged collection period. Collection of fluid is very difficult. Why? Because the viscosity of fluid makes the aspiration quite tough. Difficult to hold capillary tube at the entrance of gingival crevice, which is yet another human error. It is also difficult to remove the complete sample from the tubing. You can understand, this is practically not very possible to, this capillary tube is so small in diameter, and will you be able to collect the entire amount? Some amount will be sticking to the walls of the internal walls of the tubing. So this is the disadvantage of micropipettes. Next is the crevicular methods. Crevicular method, two scientists, Oppenheim and Skapski, They were responsible for the crevicular washing technique. Gingival sulcus is usually perfused with the help of an isotonic solution. Usually Hank's balanced salt solution is used. Two methods were suggested. One is a simpler method involving the instillation and re-aspiration of 10 milliliter of Hank's balanced salt solution in the interdental papilla, repeated 12 times to allow thorough mixing of transport solution and GCF. Transport solution is your Hank's balanced salt solution. The fluid collected then represents a dilution of crevicular fluid. This is what I was talking about earlier, that in case of micropipette method, you were able to collect an undiluted native GCF, in the same concentration we were able to collect it. But here, since we are introducing an isotonic solution of Hank's balanced salt solution, it is understood that this is a diluted form of crevicular fluid, and it will contain both cells and soluble constituents such as plasma proteins. This technique could therefore be applied either to individual interdental units or to multiple units which were then pooled. This technique was given by Skapowski et al. Next method was given by Oppenheim. What did they do? They fabricated an acrylic stent. Okay, so this method is a little complicated one, as there is you need expertise and skill to fabricate an acrylic stent. And finally, this stent, when introduced in the mouth, it should be able to isolate the gingival tissue from the rest of the tissue. The tissues are then irrigated with a saline solution using a peristaltic pump. Here is a picture of the peristaltic pump, and you can see that the lady, she has the acrylic plate which is prefabricated, and this pump is connected to the acrylic stent, and then the diluted GCF is removed. This method uses an appliance consisting of a hard acrylic plate which covers the entire maxilla with soft borders and a groove following the gingival margin. Why is this groove necessary? Because they are connected to the 4 collecting tubes. So at a time, you can collect the GCF from multiple sites with such method. The washings are obtained by rinsing the crevicular area from one side to the other with the help of peristaltic pump. What are the limitations? Limitation is that fabrication of stent is quite demanding, and it is complicated also. It has been useful only in maxillary due to the difficulties of producing. How will you fabricate stent for the mandibular arch? There is a tongue which will be hindering the stent again and again. So this is more advisable for the maxillary arch because of the difficulties of producing a technically satisfactory appliance for the mandibular arch. GCF from individual sites cannot be analyzed. Yes, all the fluid may not be recovered during aspiration and re-aspiration procedures. So there was a modification. What was the modification? And this is quite easy. You can imagine 2 injection needles, one is fitted into the other. In a way that the inner needle is at the bottom of the pocket and the outside of collecting one is at the gingival margin. Okay, so you can see that this is the inner needle which is at the bottom of the sulcus, while the outer needle that is just at the margin or the entrance. The collection needle is drained into the sample tube. Then what happens? The collection needle will be drained into the sample tube by continuous suction. So the figure 1, it shows a schematic illustration of device which is designed for intracrevicular sampling of crevicular contents. A small ejection needle is inserted into the crevice to clinically predetermine the depth indicated now by opening of collection needle at gingival margin. And figure 2, it is schematically illustrate the ejection needle which is kept in sulcus or periodontal pocket and connecting to the syringe for manual adjustment of washing time and liquid pressure. Collection needle is connected to Appendorf tube by short elastic tube. See, it is a little complicated whatever it is written here. Just imagine that there are 2 injection needles. One is fitted into the other. One is deep into the sulcus. The other is right at the gingival margin, and this is the sample is drained out with the help of peristaltic pump. Okay, so talking about the evaluation of GCF collection, use of Periotron. Before the use of Periotron, if I talked about those filter paper strips, right? Those filter paper strips, they are called as Whatman's Perio paper. They are usually wetted, and the margins, the wetted margins can be clearly demonstrated or can be clearly be visible by staining them with some kind of a dye, and the dye which is usually used is ninhydrin. It is then examined planimetrically or using a microscope or photograph or using magnifying glass. But there is an electronic method as well, which has an electronic transducer machine, and this is the electronic transducer machine which is called as Periotron. Okay, the wetness of perio paper affects the flow of electric current, and this will give you the digital readout. What do? You just place your wetted absorbent paper here, paper strip, and since it is wet, it will affect a electric current, and because of this electric current, you will get digitally what is the amount. Okay, so what are the problems which you face during GCF collection? First and foremost is contamination by saliva, blood, or plaque. Small sample amount is collected, which is very time taking also, so sampling time is quite long, and evaporation is also a significant problem. It depends on type of paper, binding of GCF protein to the filter paper and concentration of the original protein sample, so recovery from the strips is not easy. Next is composition of GCF. I hope part 1 is clear to all. So from here, we'll be discussing on next term. Composition of GCF. GCF consists of, as per Simasoni in the year 1983, he gave the composition of GCF and suggested that there are cellular elements, there are electrolytes, organic compounds, bacterial and metabolic products, enzymes, and enzyme inhibitors. So this is a mnemonic which I prepared. This is PDAF Nox. So BDF Nox. Okay, if you can remember, B is bacteria, D are degradation products, E is epithelium, F is fibroblasts and neutrophils, O C are the osteoclasts, and S are the serum. So gingival crevicular fluid is composed of substances derived from serum, leukocytes, bacteria, activated epithelial cells, connective tissue cells, and bone cells. Okay. Important question arises that what is the permeability of junctional epithelium and sulcular epithelium that permits GCF to flow into the sulcus? Substances like albumin, histamine, phenytoin, endotoxin, they were known to cross the sulcular epithelium, and experimentally substances like Indiana ink, saturated iron oxide, fluorescein, they were found to cross the sulcular epithelium. Thus, it was indicated permeability of sulcular epithelium was up to molecular weight of 1000 kilodalton. Yet another important point. Armitage also gave the composition. Again, he suggested that there is a presence of leukocytes, proteins, enzymes, tissue breakdown products, inflammatory mediators, cytokines, etc. Let's discuss about the composition in detail. If we talk about the cellular elements, cellular elements include bacteria, desquamated epithelial cells, leukocytes. Electrolytes are the inorganic products. They include the potassium, sodium, calcium, phosphate, magnesium. Organic compounds include carbohydrates and proteins, glucose, hexosamine, and hexuronic acid. These two compounds are constantly found in GCF. Which one? Glucose, hexosamine, and hexuronic acid. Glucose in GCF is usually 3 to 4 times higher than that of serum. It is also an important bio-ghost question, that what is the amount of glucose in GCF and compared to the serum, usually 3 to 4 times higher, and it is also contributed to the local microbial activity. Total protein content of the GCF is much lesser than that of serum. Bacterial products present in GCF include lactic acid, urea, hydroxyproline, endotoxins liberated by gram-negative bacteria such as AA, P. gingivalis, cytotoxic substances like hydrogen sulfide, and antimicrobial factors. So these are some of the components that we find in GCF. What are the enzymes? Enzymes are acid phosphatase, alkaline phosphatase, lysozyme, lactoferrin, PGE2, collagenases, etc. Coming to the clinical significance of GCF. What is the established clinical significance of GCF? As we have already seen that GCF is an inflammatory exudate used to determine the inflammation of gingiva, mostly as subclinical gingivitis indicator. That is, subclinical gingivitis can be estimated by the presence of GCF. The amount of GCF is greater when inflammation is present. So sometimes, we can say, yeah, that amount of GCF is directly proportional to the severity of inflammation in that area. Next question arises, what causes GCF amount to fluctuate? So factors responsible for GCF fluctuations. First is your circadian periodicity. Circadian periodicity means there is a gradual rise of GCF from 6 a.m. to 10 p.m. and then there is a decline of GCF. Next is your sex hormones. Sex hormones, especially female sex hormones, what do? They increase vascular permeability. Because of the increased vascular permeability, it is but natural that the production of GCF will also be increased. So what are the conditions where you will see fluctuation of sex hormones? Pregnancy, ovulation, and contraceptive uses. So during pregnancy, as suggested by Low in the year 1965, during menstrual cycle, Mulliman in 1948, and at puberty by Sutcliffe in 1972, there is an exacerbation of gingivitis. Therefore, there will also be an increase in the GCF amount. According to Linde et al. In 1969, female sex hormones, they cause vascular permeability, as I just told you. Next is gingival fluid in diabetic patients. According to Ringelberg et al. In 1977, higher flow rate of gingival fluid in diabetic patients were reported compared to healthy counterparts. Investigators agree that the exudates collected from the diabetic patient contain significantly more glucose. And I just told you that the amount of glucose is usually 3 to 4 times higher than that of serum. Yet another MCQ based question. Coming to the drugs in gingival fluid. Okay, two important drugs, which are your tetracycline and metronidazole. Meanwhile, two more factors I miss on. Sorry, first one is the mechanical stimulation, which is also responsible for increasing GCF. That GCF amount can be increased by providing stimulus through chewing or by vigorous gingival brushing. Smoking also causes an immediate but transient increase in GCF flow. So let's talk about drugs here. So Stephen et al. In the year 1980 measured the concentrations of ampicillin, tetracycline, erythromycin, and clindamycin in serum, saliva, and gingival fluid after a single oral dose administration. Gingival fluid antibiotic concentrations were equal to those found in saliva. They were, however, always much lower than the concentrations found in serum. So these drugs can be used in periodontal therapy, and one of the most common drugs, to have been found concentrated in GCF include tetracycline and metronidazole. Therefore, the local presence of these drugs can be used as an advantage to treat periodontal disease. Okay, this slide, sorry, it was missing earlier. So we've already discussed this slide, that the mechanical stimuli. The effect of chewing was investigated by Brill et al., and the effect of vigorous toothbrushing was suggested by Fry et al. So the amount of gingival fluid was shown increased significantly under the influence of chewing and vigorous toothbrushing. Coming to periodontal therapy and crevicular fluid. The effect of repeated prophylaxis on plaque accumulation and gingival fluid flow. They found that the fluid flow decreased 1 week after the first prophylaxis and then slowly returned to pre-treatment values. After a second prophylaxis, the lower levels of fluid were sustained for longer periods of time. Thus, we conclude that through the biomarker discovery process, new therapeutics have been designed, linking therapeutic and diagnostic approaches together, leading to more individualized and targeted treatments for oral health. Thus, the origin, the composition, and the clinical significance of GCF have significantly helped our understanding of the pathogenesis of periodontal diseases. These were my references. Thank you.